Welcome to BioLens Learning YouTube channel.
First, prepare a thin smear on a clean glass slide. Allow it to air dry completely. This kills the organism to the slide. Your slide is now ready for staining. Now flood the smear with Carbol Fuchsin. Heat gently until steam rises. This allows the stain to penetrate. Acid-fast cells will appear red. Step 3. Cool and rinse. Allow the slide to cool. Rinse gently with tap water. The slide now appears lighter. Step 4. Decolorize with acid alcohol. Continue until the smear appears pale pink. Be careful not to over-decolorize. Non-acid-fast cells are now colorless.
Step 5. Rinse the slide gently with tap water. This should take about 1 minute. Rinsing removes the acid alcohol and stops decolorization. Now, we are ready for counterstaining. Step 6. Counterstain with methylene blue. Flood the smear for 30 to 60 seconds. Then, rinse gently with tap water. Acid bacteria appear red. Step 7. Final rinse. Rinse the slide gently with tap water. This removes excess counterstain and prevents crystal formation. Acid-fast appear red. Step 8. Air dry. Allow the slide to air dry in a dust-free area. Acid-fast bacteria appear red or pink. The slide is ready for microscopy. Step 9. Examine the slide under the microscope. Use the oil immersion objective against blue cells. Scan the entire smear systematically. Now you can interpret the result.
Step 10. Results. Red or pink bacteria indicate a positive acid-fast stain, non-acid-fast material. Staining completed successfully.
Thanks for watching. Subscribe my channel.